treatment group did get about half that, and the control group more or less maintained their diet. During the CALERIE trial, there were blood draws at 3 time points. That blood is in the freezer in Vermont. You can get it. I'll talk more about it in a minute. But a number of assays were also performed of that blood and are available currently on the website. CALERIE clearly had effects on the weight of participants. Again, something that Bill reviewed. What we're interested in testing here is this hypothesis that the ad libitum group is going to continue to age at kind of a normal rate over these 2 years of the trial, and the CR group is going to age at a slower rate. So we're going to measure this construct of biological age using a couple of off-the-shelf methods that can be applied to data that are already available on the website. So I literally went to this website, which was slightly less attractive at the time, and downloaded analysis-ready data files to my computer, and with a little bit of support from Carl and Bill and Manju, was able to interpret these files sufficient to compose a data set. And here's what we did. We took algorithms to quantify biological aging from clinical chemistries that had been published by Morgan Levine, now at Yale in the Department of Pathology, and Alan Cohen at the University of Sherbrooke in the Department of Medicine, to quantify the rate of aging in these individuals. And we had to tweak these algorithms slightly based on the available biomarkers in CALERIE. And so we did a little validation exercise to make sure that what we were doing was reasonable. We used data from the National Health and Nutrition Examination Surveys, so the CDC's NHANES study, which have a lot of clinical chemistry data, chronological age information, and as I'll show you, information about disability. We matched the years of the NHANES that we were analyzing to the years of the CALERIE trial so we could wash out any period effects in terms of what people were being exposed to in the general public health and medical environment. And so the question is, we can definitely predict the chronological age of these samples using these methods. That's what they were designed to do. The question is, can we predict something more interesting? Can we predict the increase in risk for, let's say, disabling disease that we would like to slow or prevent with geroprotective therapy? So we're going to use a simple self-reported measurement of disability, activities of daily living scale, in the NHANES to do this. And what I'm showing you here is just that as people in the NHANES get older or older compared to younger people in the NHANES, have more disability, right? So disability goes up. And so what we want to know is, can we explain that kind of age-dependent increase using our biological aging algorithms? So we trained the algorithms in the 2007, 2008 NHANES. We're now testing them in the 2009, 2010 NHANES, so completely independent data sets. These are people aged 30 to 75 years. And what I'm showing you here is that if we take people who are just 50 years old but who vary in their biological age essentially recovers the chronological age distribution of the prevalence of disability in the NHANES, all right? So these measures of biological age explain the age-dependent increase in risk for disability in this data set. And looked at another way, if we fix their biological age and allow them to vary in terms of their chronological age, we see no change. So essentially, the information content in chronological age with respect to disability is recovered by the algorithm. Okay, so we have some evidence that we're measuring this biological age construct. And there's published evidence that these algorithms predict mortality independent of chronological age and prospectively predict disease and disability independent of chronological age. So we have a measurement that we think is interesting. And what we'd like to do with it is now take that algorithm and put it to work in the CALERIE study. So what I'm showing you here is a scatter plot at baseline of all of the CALERIE samples. The blue dots are boys, the pink dots are girls. And I'm showing the correlation between their chronological age and their biological age as estimated by these algorithms, and in particular, the Morgan Levine's algorithm here. The correlation is very high, it's 0.9. We can predict the chronological age of our study members very well. You can see that most of the dots are falling below the line of unity here, so they're a little bit younger than the NHANES data would predict they should be based on their physiologies. I would guess that's healthy participant bias, but there are, of course, other variations in assay chemistries that could potentially produce something like this. I think what we're looking at is healthy participants versus the general population. Okay, so now the real test is, okay, but does it change as we follow these people over time? And so what we see here, what I'm graphing on the y-axis is change in this measured biological age relative to baseline. So everybody's change is 0 at time 1. And if this was working perfectly, the change would be 1 at time 2 and 2 at time 3 over the 2 years of the trial. That's not quite what we got. There's about a 3/4 of a year biological age increase per 12-month interval in the CALERIE trial in the ad libitum group. So for people who did not undertake caloric restriction, they aged more or less at the rate we expected them to, although again, consistent with what we saw at baseline, a little bit slower than the NHANES data would suggest. So then, the test was, is that rate slowed in caloric restriction? And again, this is intent to treat. So these are just people randomized to CR, irrespective of what their achieved dose of CR was, more or less didn't change in their biological age from baseline to follow-up. So by this test, randomization to the CR condition in the CALERIE trial slowed the rate of biological aging. And we see a consistent result using the homeostatic dysregulation algorithm as well, although in that case, the specific result is that homeostatic dysregulation is roughly maintained across the trial in ad libitum and actually improves in CR. So people maybe get younger as measured by that test. In general, though, both methods suggest that caloric restriction slows the rate of biological aging. Okay, so what we would like to now do is try and take this a step sort of down the ladder from physiology to cellular level changes, these hallmarks or pillars of aging. And of those, the most tractable for sort of human epidemiological study at this point appears to be DNA methylation changes that occur with aging. Broadly across the genome, as we get older, methylation levels decline, and the theory is that this may be related to broad genomic dysregulation that's related to the process of aging. But at the level of specific nucleotide sites along the genome, DNA methylation goes up in some cases and goes down in others in a surprisingly regular way. So regular that people have been able to parameterize algorithms that they call clocks that can predict the age, the chronological age of a tissue sample based on a few dozen or a few hundred of these DNA methylation marks, these chemical tags on the DNA. So Steve Horvath has made this an extremely easy-to-use method, producing an online accessible tool, and there have now been dozens of studies applying these epigenetic clocks. It's a very robust result. They predict the chronological age of a tissue. There's some evidence that they can prospectively forecast mortality in older adults. The first meta-analysis has been published. So this is where we targeted our inquiry. And so what we did was, again, we went back to the CALERIE website, and we had kind of an inside line through the external scientific committee. But we prepared a proposal. We said, we'd like to do this study. Could we have some of the blood from the baseline, or some of the DNA from the baseline collection? And this is what we'd like to do with it. And they said, sure, and with actually a tremendous amount of effort from Bill and others. I shouldn't say a tremendous amount of effort. It was really very easy to do. But there was a material transfer agreement involved. And for those of you who haven't undertaken that process of getting tissue from one place to another, there are many forms to fill out and so forth. And so all that was accomplished. And the tissue was brought to Duke, where it was given to Karen Abramson and her core at the Duke Molecular Physiology Institute, where they ran these 186, or I guess there were 192 total DNA samples with the Illumina EPIC chips. So this is 850,000 roughly CPG sites that are quantified by the chip. And those data were then passed off to my colleague David Corcoran in genomics and computational biology, who did the bioinformatics, essentially data quality control and some processing for us. And it turned out that the data were actually in remarkably good shape. So all 192 samples passed quality controls. The 6 duplicates were nearly perfectly correlated with in person. There was a slight hiccup. For those of you who are working with the EPIC chips, it turns out Illumina rolled out 3 different flavors of the same chip without telling anyone. And there are slightly different numbers of probes on all the chips. So there was a little hiccup in the data processing that slowed us down, which is why we didn't get this data until last week. But here they are, hot off the presses, and now this problem has essentially been solved by the publicly available bioinformatics pipeline that is pushed to people who want to use these Illumina chips. So it's not a problem anybody has to worry about. We're going to do two things with these data. I'm going to show you some results from the Horvath Labs website, mostly by way of data quality control, and then I'll show you some results that speak to this question of whether caloric restriction may slow biological aging as measured by these cellular level parameters. And again, we only have epigenetic data at this point at baseline, and you will all have access to that data too in the coming weeks and months, depending on how long it takes us to finally wrap this data site up and post it. Okay, so as advertised, the epigenetic clocks tick. The correlation with chronological age is about 0.9. You can see in this particular case, the dots are mostly falling above the line of unity, and this is something that we know of other labs experiencing as well, that epigenetic clocks computed with Illumina 850K data seem to be running a bit fast, so to speak, so that people tend to be, on average, older. For our purposes, what's most critical is that the variation be consistent and not that the absolute estimate be correct, but that's something worth being aware of. Other comforting bits of data, the epigenetic data can accurately predict the sex of the samples that were given to them. That's reassuring. And we can compare the estimates of the cell counts generated from a widely used algorithm applied to the epigenetic data with the complete blood count data in CALERIE. And for some of you, correlations in the 0.4 to 0.6 range may not be very impressive, but this is entirely consistent with what we've seen in other data comparing Hausman algorithm estimates of cell counts with complete blood count data.